In the last topic, we had seen that pollination. We had dealt with pollination by biotic agencies and abiotic agencies, of which I had mentioned that pollination by insects is known as entomophily, but we did not go through inside the topic. See, a diagram is being given in your chapter by pollination by insects, and in which a flower is being given and a honeybee is there in that flower. That means we can take any flower, because many of the flowers are being pollinated by insects, but I have considered one such flower as salvia because it follows the liver mechanism. What is a liver mechanism? If you see the flower in salvia, salvia are violet colored flowers. The same flower can be seen in Andamans, like it is a dark blue in color and it occurs in white color also. It is a climber. So if you see the flower, it has got two types of anther. One is the fertile anther in salvia, and this is the unfertile anther. Here is the nectar. Here is the nectar. So, if the insect comes, the insect lands here. If you see the insect, it will land on this part of the salvia, this petal. When the insects land on this salvia petal, what happens? Since you see it is of unequal length, so it tries to gain entry because it has to take pollen from here. When it has to take pollen from here, so when it enters, it lands on the unfertile anther. It will lie on the unfertile anther. If you see, both the anthers are being connected by a filament. It is a long filament, and in salvia follows protoandry. Salvia follows protoandry. That is andrecium. Andrecium matures first. Salvia follows protoandry. So what happens? When it is gaining entry towards the pollen grain, it lands on this pollen. Okay, it will land on this anther. This anther, if you see, it has got a long filament. It will come and it will dust on the body of the insect. It will come and lie because it is gaining entry. So it stabilizes. When it stabilizes, पैर रखकर इसको बराबर जाने की कोशिश करेंगे इंसेक्ट. जब ये इसमें छोटा सा ये पेटल है, वे इसमें जब जाएंगे तो ये जो एंथर है ये आके इसके ऊपर डस्ट होएगा. सारा पोलन ग्रेन ये इंसेक्ट के ऊपर डस्ट होगा. फिर ये इंसेक्ट पोलन नेक्टर लेकर ये फ्लावर से उड़ जाएगा. जब वो दूसरे फ्लावर में जाएगा, when it goes to the other flower, what happens? Here the stamen is there. Here the pistil is there. Another flower. As I said, it follows protoandry. तो protoandry बोलने से andrecium will mature first. जब andrecium mature हो के चला जाएगा तो ये andrecium dehisce हो जाएगा, shrivel हो जाएगा, wrinkle हो जाएगा. तो दूसरा फ्लावर में जब जाएगा तो उसका पिस्टल रेडी है. तो जो इंसेक्ट जब ये नेक्टर लेने जाएगा तो सेम वे जैसा ये एंड्रोशियम आके उसका बॉडी में लगा, उसी तरह ये पिस्टल आके इंसेक्ट का बॉडी में टच होगा. When it will touch the body of the insect, जैसे ही टच होगा तो उसका बॉडी में ऑलरेडी पोलन ग्रेन था, तो वो ये स्टिग्मा में जाके अटैच हो जाएगा. और what is pollination? Pollination is the transfer of anther, pollen grains from anther to the stigma. तो pollen grain transfer हो गया और hence pollination हो गया. So this is following the liver mechanism. The example given in your book is by insects. There is no such mention as which flower. I have particularly taken salvia flower just because it is following the liver mechanism. Because majority of the flowers are chasmogamous flowers. खुला रहता है उसका stigma, तो उसमें कुछ problem नहीं होती है. It is a peculiarity, that's why it is being mentioned. And if you remember, in the last class, I had explained about hydrophily in Vallisneria. Vallisneria is very important. It always comes in the exam, either in a 3 marks question or in a 2 marks question. I had drawn the diagram of Vallisneria. If you remember, like this I had drawn, it has got spiral and here was the female flower. See, when I was mentioning, I mentioned in the same plant, like the female flower also I mentioned in the same flower, the male plant also I mentioned in the same flower, just for explanation. But that doesn't mean that both belong to the same flower. It is not a bisexual plant. Vallisneria is not a bisexual plant. I had explained in the diagram because it becomes much clearer, but if you see Vallisneria, male is present in a different flower, different plant, female will be present in a different plant. Rest all the mechanisms are same. But you should remember that if I have drawn that way, that means it is belonging to the separate plant. This is a separate plant, that is the female plant, and this is the male plant. They will be present in the water. They will present side by side, but they will present it will follow the female plant, female flower. This will follow the male flower or the male plant. Again, it has got the male is the inflorescence. Majority of the pollen grains are present. It is above the water. What I had explained previously. This is a very important topic, hydrophily in Vallisneria. Now pollination is being completed for you. What were the various outbreeding devices? What were the various outbreeding devices? That has been dealt with, how the plant itself does not allow for self-pollination to take place. One such example is the pollen-pistil interaction. One such example is the pollen-pistil interaction. Now we see that in nature we have already said that it follows the abiotic agencies also, that is by water, by wind. So if by wind, anemophily, that is pollination by wind. Suppose if pollination is taking place by wind, so there are so many flowers in the environment, pollen grains could fall in every flower. So why the pollen grain of that plant does not pollinate this flower? That question will arise in the mind. So there are two types of species, that is interspecies and intraspecies. That is pollination, interspecies pollination will occur within the species and within different species, and intraspecies means similar species. Interspecies between two species, intraspecies between the same species. एक species के अंदर कितने plants हैं उसके वजह से ये pollination होगा. लेकिन ऐसे में तो pollination होना चाहिए. एक ही स्पीशीज के अंदर आपका कितना जीनस और कितना रहता है, स्पेसिफिक रहता है. एक जीनस के अंदर कितना स्पीशीज रहता है, स्पीशीज के अंदर कितना प्लांट्स रहते हैं. तो ये जब एक ही स्पीशीज को बिलोंग करते हैं तो ये सारे फ्लावर्स को आपस में पॉलिनेट होना चाहिए, बिकॉज दे बिलोंग टू द सेम स्पीशीज. बट व्हाई दे आर नॉट पॉलिनेटिंग, व्हाई दे आर सो स्पेसिफिक, दे आर नॉट पॉलिनेटिंग. कायदे से it should pollinate because they belong to the same species, they should pollinate, but majority of the flowers are not following intraspecies. Within the same species also they are not allowing pollination. Why? Why they are not allowing within the same species? If you see that this is the flower, so I'm just drawing like that, it's a flower, suppose, okay. So this is the stigma. If I take this is the stigma. Okay, stigma, style, ovary, stigma, style, ovary, and here are the stamens. Here are the stamens. So if a pollen grain is landing on the stigma, the pollen grain is landing on the stigma, it should germinate. Why all the pollen grains, if it is within the same species, belonging to the same species also, it is not germinating? Why it is not germinating? See, this is the stigma. We had already seen in microsporogenesis how the pollen grains are germinated. See, when the pollen grains are forming on the stigma, it can fall as the two-celled stage. It will fall in the two-cell stage, that is vegetative cell, generative cell. When it is just as it is following on the stigma, what happens is that when it follows in the two-cell stage, already the two male gamete are being formed. When it falls on the stigma, it falls in a two-cell stage. If it is falling in the two-cell stage, what do you understand by the two-cell stage is that this vegetative cell is nowhere in picture. That is being totally consumed, and this generative cell, it is forming the two male nuclei. By utilizing all the vegetative cell, it has formed the two male nuclei. This two male nuclei will fall on the stigma. Sometimes it follows the two-celled stage. Pollen will fall at the two-celled stage. And sometimes it will follow the three-celled stage. It will follow the three-celled stage. What is three-celled stage? That means there are the two male nuclei and a remnant of the vegetative nuclei. थोड़ा सा vegetative nuclei, थोड़ा सा part बचेगा just उसको और नरिशमेंट देने के लिए. तो दो टाइप से फॉलो करेगा, टू सेल स्टेज एंड थ्री सेल स्टेज में पोलन ग्रेन फॉल करेगा. एंड वी नो, वी हैड सीन हाउ द पोलन ग्रेन्स आर देयर. पोलन ग्रेन का पता है हमें, इट इज द आउटर एक्साइन, दैट इज द हार्ड पार्ट, एंड इट इज द इनर इंटाइन. इंटाइन जो है आपका है ये मेम्ब्रेनस है, इट इज अ मेम्ब्रेनस इंटाइन, और उसका अंदर टू मेल न्यूक्लियाई. तो जब वो स्टिग्मा के ऊपर फॉल करेगा तो एक्साइन तो रहेगा नहीं, एक्साइन के बीच बीच में ओपनिंग्स रहता है. देयर आर जर्म पोर्स, यस, देयर आर जर्म पोर्स इन द पोलन. तो व्हेन इट फॉल्स ऑन द स्टिग्मा, द जर्म पोर विल कम आउट. So there are so many germ pores in the pollen grain. ऐसा नहीं है कि एक ही पोलन ट्यूब निकलेगा, एक पोलन से एक ही पोलन ट्यूब निकलेगा. समटाइम्स एक पोलन ट्यूब निकलता है और कभी कभी बहुत सारा पोलन ट्यूब निकल सकता है. यहाँ से निकल सकता है, यहाँ से बहुत सारा पोलन ट्यूब निकल सकता है, लेकिन एक ही पोलन ट्यूब को एंट्री मिलेगा. ठीक है, भले ही एक पोलन ट्यूब निकले या 4-5 पोलन ट्यूब निकले, लेकिन इफ इट इज कंपैटिबल, एक ही पोलन ट्यूब को एंट्री मिलेगा within the style. So what is pollen pistil interaction? This is how it is taking place. So what is pollen pistil interaction? We have seen how the pollen will form, which stage it will form. Now we will see. This is the stigma, style, ovary. See, ovary here, this is the style. It has got some enzymes and acids, Hybridisation and nutritive tissues. It could be anything, cellulase, what all which can provide nutrition to the developing pollen grain. So this is present in the style region. There are some chemicals which are present in the stigma. There is some chemical which is present in the stigma. What is that chemical that will identify? See here, this is the ovary, this is the ovary region. Okay, suppose the ovary has got S1, S2. Okay, suppose the ovary has got S1, S2. So many pollen grains fall. There are many pollen grains which will follow S1, S2, S3, S4. So the stigma will recognize the compatibility. The ovary, what it is having? It is having S1. So of the many pollen grains which are following on the stigma, you can see this is the S1 and here is the S2. So these two S3, S4 will not be allowed. It is only the S1 which will gain the pollen tube, from which the pollen tube will emerge. And this pollen tube, it will again gain entry. When it gains entry in the styler region, for its length also, the pollen tube length, it will gain entry. The pollen tube length, it will also be dependent on the style. The style will provide nutritive tissue. So it will come and it will reach the ovary and here it will fertilize. Similarly, if it is S2, then also it will fertilize. Sometimes it is very, very specific. It will only be S1. This is also S1 and this is also S1. It is be so specific. That is, it recognize the pollen grains. So it will not allow any other pollen grain to germinate. That is the pollen pistil interaction. The pollen is being recognized by the pistil. Pollen is the male reproductive part of the flower, pistil is the female reproductive part of the flower, stamen is the male reproductive part of the flower, which consists of the pollen also, and this is the female reproductive part of the flower. So here fertilization will takes place when it will reach the ovary. So when it reaches the ovary, how does fertilization takes place? How does fertilization takes place? We had already seen the female gametophyte. This is a female gametophyte, micropyle, chalaza, embryo sac. So the pollen which has been produced earlier, it has to reach here, embryo sac here. Here is the egg, this is the egg, and these are the synergids. And here antipodal cells. We had done already. Here is the polar nuclei. Here is the polar nuclei. So, of the two male nuclei which was present in the pollen tube, one will come through the micropylar. One more mention is that it can gain entry through three ends. Pollen grain can gain entry through three places. One is through the micropyle. If it is through the micropyle, it will be known as porogamy, or through the chalaza. If it is, then it will be known as chalazogamy, or through the integuments, then it will be known as mesogamy. There are three ways of entry of the pollen tube or the male nuclei towards the egg. So, either it takes place through porogamy, or it takes place through chalazogamy, or it takes place through mesogamy. It will, after all, it will come and fertilize the egg, and it will be fertilize and it will form the endosperm with the secondary nuclei, polar nuclei, which we have already seen. So, one of the male nuclei of the two which was being produced in the pollen tube, two male nuclei, one of the male nuclei fertilizes with the egg and it forms 2N, that is, this is forming zygote, egg plus one male nuclei fertilizing, and it is forming the zygote, and another male nuclei fuses with the polar nuclei, another male nuclei fuses with the polar nuclei, and it is forming the 3N structure, or it can also be known as the endosperm. It can also be known as primary endosperm nucleus, PEN. So, two times it is fertilizing. Once it is fertilizing with the egg and it is forming the zygote, another it is fertilizing with the polar nuclei and it is forming the endosperm. So, fertilization takes place two times. That's the reason it is known as double fertilization. And why it is known as triple fusion? 2 already plus 1, 3N. That's why it is known as triple fusion. Either it will be known as double fertilization or it will be known as triple fusion. By this way, the zygote is forming and the endosperm is being formed. Now, if we see, till this we have made the zygote embryo. Zygote will develop into the embryo. How the zygote will develop into the embryo? Before that, one more thing which we have to see is, see, we had seen how pollination takes place and how the nature itself undergoes outbreeding devices just to prevent self-pollination. Similarly, what we are following artificially, we are controlling the pollination. We can also known as artificial hybridization. That is, we are pollinating, we are dusting our desired pollen grain on the stigma of a plant. So, we are doing it artificially. So that is known as artificial hybridization. Why it is known as hybridization? Because in nature what should have been produced, we are not happening, we are not making it happen. We are dusting our desired pollen grain artificially to make a hybrid plant. That's why it is known as artificial hybridization. अपने मन से जैसे बनाते हैं. तो once they are following a particular technique, 3 steps it is taking place. Artificial hybridization, that is emasculation, bagging and tagging. Emasculation means removal of anther. Emasculation means removal of anther. That is, if it is a bisexual plant, we have to remove the anther. If it is a bisexual plant, we have to remove the anther. 2 plants are separate. That means anther is present separately and the female pistil, this is present separately in separate plant. If it's a monoecious plant, that is both the sex organs are present in the same plant, female and male. That time we have to remove the anther. How the anther is being removed? You see there are forceps which are present when you perform practicals. There are a pair of forceps. Through the forceps from the flower, suppose this is the flower, this is the stigma, style, ovary, okay. This is the flower. Here anthers are present. So this anther is being removed. And after the removal of the anther, you are removing anther from the flower, but still the plant is exposed to the air. So removal of anther is known as emasculation. In a monoecious plant, we have removed the anther, but still the stigma is exposed. So what is being done? Bagging is being done. What is bagging? We cover the flower with a polythene bag or a paper bag, translucent paper bag. We will cover the flower, okay, we'll cover the flower with a paper bag or a polythene bag, translucent. Okay, that is known as bagging. When you cover the flower with bag. Why we are covering the flower with bag? Because the stigma is still exposed. So now there are no chances of pollination to take place. We have removed the anther, that is known as emasculation. We have covered the flower, that means we have covered the stigma, that is no foreign pollen grain also will not fall on the flower. So prior to bagging, before we do bagging, what we will do? In bagging, we will cover the bag. As I said, we will cover the bag, but before covering it, we will dust our desired pollen grain. That's why I said artificial hybridization. Which kind of flower we want? That kind of flower, a particular superior quality pollen grain, we will dust on the stigma. After we dust on the stigma, then we cover the flower with the bag. Then we cover the flower with the bag. That is known as bagging. I repeat it again. What is artificial hybridization? Artificial hybridization means we are preparing our own plant. That is artificially, we are dusting our desired pollen grain. How? It is being carried out in 3 steps, that is emasculation, bagging and tagging. Emasculation means removal of anther. Why removal of anther takes place? Because if it's a monoecious plant, both the sex organs will be present in the same plant. So in that way, we have to remove the anther. So removal of anther is known as emasculation. Again, bagging, after removing, we will dust our desired pollen grain on the stigma. Now the stigma has got our desired pollen grains. That is, now pollination will take place. When the pollination has taken place, when we have dusted, we will cover the flower. Because we have placed our desired pollen grain, we don't want any other foreign pollen grain to land on the stigma of that flower. After that, it will be covered, then it will be tagged. What is tagging? Tagging means we are preparing a paper slip. We are preparing a paper slip so that we are giving the names, like name of the plant, which pollen grain, which species it is belonging, and what plant species we have dusted, which plant we have dusted on the pollen grain, the date, the time, everything will be done and it will be tagged. This paper tag will be kept in the plant. So the pollinator will know, or the plant breeder will know at which time it was done. And after some time, the pollination will take place, the flower will develop into fruit. So hence, this is how artificial hybridization takes place. We had seen how the embryo was formed, that is the 2N, and how the endosperm, which is formed, that is 3N. The formation of embryo, how the dicot embryo and how the monocot embryo will form, that I'll discuss. Prior to that, what is the nutritive tissue which will help in the formation of the embryo, in the development of the embryo, that is known as endosperm, because it is the endosperm which is the nutritive tissue on which the embryo will survive. So how the formation of embryo will take place? Prior to that, we will see what is an endosperm, what are the types of endosperm. So endosperm, we know it is a nutritive tissue on which embryo will grow. Embryo will take this endosperm and then further development of embryo will take place. There are three types of endosperm, that is nuclear, then cellular and the halobial. In some books there are the mention of ruminant endosperm also. In some book there is the mention of ruminant. Ruminant means which is leftover. So hence majority of the books don't deal with ruminant endosperm. We deal with only three endosperms, that is the nuclear endosperm, cellular endosperm and the halobial endosperm. How the endosperm is being formed? What happens to the endosperm? How it is formed? See, when the embryo is formed, after that we know that the endosperm is also found in the embryo. So how the endosperm is formed? In the nuclear type, we see all the nuclei, free nuclei, they will form, they will divide by mitotic division, all the nuclei. It will continue to form in the nuclear endosperm, it will continue to form, and in the center if you see, there is hollow. That is, no cell wall is formed, no cross walls will form. Hollow means, here hollow means, that doesn't mean that it is completely hollow, it means that there is no wall formation. So it is free nuclear, that's why we also say it as free nuclear. Either you say it as nuclear or you can also say it as free nuclear, because it is free. That is the peculiarity of the nuclear endosperm, that which you are seeing in coconut. Next is your cellular endosperm. In cellular endosperm, what you will see, as and when the nucleus will form, suppose two nuclei, it is dividing mitotic, as and when the nuclei will form, the cross walls will also start forming. The nuclei will form, the wall will form. Nuclei will form, wall will form. So after some time, what you see, it is completely cellular. All cell, inside the cell nuclei is formed. So it is completely cellular. This type of endosperm. This type of endosperm, it means it will be very hard because cell wall is being formed. What you see in areca plants. So such kind of endosperm. Third is the halobial endosperm. Halobial endosperm is a combination of nuclear endosperm as well as cellular endosperm. In halobial endosperm, what you see, in the micropylar region, okay, in the micropylar region, what you will see, there are some free nuclei, it is formed. Free nuclei. Micropylar region, it will be free nuclei. And in the chalazial end, it will form cellular. At the chalazial end, cross walls will be formed. And in the micropylar end, free nuclear will take place. And in the chalazial end, cross walls will be formed. That is the peculiarity of the halobial endosperm. That is, it is a combination of free nuclear and cellular endosperm. And hence, in the chalazial end also, very few cellular endosperm will be formed. Very few cells will form cellular endosperm. Rest all will form the free nuclear form. That is the peculiarity of the halobial endosperm. But majority what we are seeing is the free nuclear type of endosperm. What you see in the coconut, this is how endosperms majority are falling in the angiosperm plants, although there are 3 types of endosperms. So we have dealt with the embryo endosperm formation, we have seen how pollination takes place, we have seen that how pollen pistil interaction takes place. In the next chapter, we will deal with the formation of embryo.